Hi, everybody. So we are on chapter 17, blood. And in fact, I wanted to record this part of the PowerPoint and this part of the lecture because we had just finished discussing in our previous lecture chapter 17, blood. And in fact, we ended up talking through most of the erythrocytes or red blood cells. So I want to take this opportunity to then discuss leukocytes, right? So I want to begin discussing that, and then I hope to discuss also platelets as well, and then a process of clotting, if you will, that occurs in blood. So we'll begin on slide 68, as you can see here on the screen, leukocytes, which is section 17.4 in the book. So when we look at leukocytes, also known as white blood cells, and so they're the only formed element that we have in blood, which is consisting of red blood cells, white blood cells, and platelets. That's the only one that is complete, and it does have a nuclei and organelles. The only one there. So red blood cells do not, because they become anucleated in the process of forming into a mature red blood cell. And of course, platelets do not, because they are actually pieces of a more of a whole cell. So white blood cells are in fact the only cell that's complete, which means it does have the nuclei and the organelles as a complete cell would have. So 1% of the total volume of blood is composed of white blood cells, and that's per microliter. Its function, general function, defense, in particular. So they, in fact, play a role in immunity, right? Which is going to be a conversation we're going to have after our exam here that covers the endocrine system and the cardiovascular system. We'll begin to talk about, in fact, the lymphatic system and the immune system. And we'll go back to white blood cells because they play a role essentially in that. So if you look at it, they really do play a dual role. They're part of both systems, cardiovascular system, but also the immune system. So their process of fighting against disease or protecting us from disease uses the process of actually leaving the capillaries, and they have this process of doing this, and we call that diapedesis. Diapedesis is the actual process by which white blood cells can squeeze through capillaries, the capillary wall, in fact, and then go to the source of any disease or infection. That could be the skin layer, in fact, right? So they go through tissue, through amoeboid motion, and positive chemotaxis, which tells us then, positive chemotaxis tells us that a cell, for example, will be in a white blood cell. This would be my white blood cell, can actually, in fact, go to the source of, say that's disease, can go through the source of disease because they use chemical releases, and they attract to those chemical releases. And so they can actually follow basically the trail to disease, in fact. And that's what you see there in white blood cells. We call this positive chemotaxis, going towards the source, positive. Okay, so when we talk about these process, and keep in mind, you can see the builded print here, and that means a term that you need to be familiar with in the text. Leukocytosis. Leukocytosis really talks about a designated white blood cell count over 11,000 per deciliter. And you can see that our range here, in fact, is 4,800 to 10,800 white blood cells. Leukocytosis is over 11,000. So we have an increased amount of white blood cells. That's what we're seeing here, an increased amount of white blood cells. This is usually as a result of an infection. So that's what we usually see in the human body, a increased white blood cell count. White blood cells, in fact, or leukocytes, fall under two categories: granulocytes versus agranulocytes. Granulocytes contain granules, and the ones that are considered white blood cells that are granulocytes include the neutrophils, designated by an N here, eosinophils, which is designated by an E here, and basophils. Okay? So those are your granulytic granulocytes. Your agranulocytes do not contain granules, but they are considered agranulocytes, and those, in fact, are your lymphocytes and your monocytes. So here's the monocyte, the mnemonic, to remember: never let monkeys eat bananas. Okay. So this is a way to remember all of your leukocytes. Okay, let's move on here. So what you're looking at here is the formed elements. So if you were to take a vial of blood and take that vial of blood from the patient and spin it down or put it in a centrifuge, you in fact would see at the bottom, filled with the bottom of the vial, erythrocytes. The middle portion, which I'll designate in white here, well, the white doesn't come out too well, let's say green, would be leukocytes, and then the top portion would be, in fact, platelets. Okay. And again, this is what you would see, for example, if you were to take a vial of blood and then take that vial of blood and spin it down. You see the erythrocytes, the leukocytes, and the platelets. Okay, so you have more erythrocytes per volume of blood than you would have leukocytes, and platelets are pieces of cells. They're less dense, so they actually are found at the top of the vial here. And so what you're seeing here, in fact, is a breakdown of all of those leukocytes, in fact. So out of those leukocytes, what volume of those particular types of leukocytes would they belong to? So a good majority, or a vast majority, of your leukocytes are in fact neutrophils, 50 to 70%. Small percentage of eosinophils, and basophils are even smaller percentage. The second largest amount by volume of white blood cells are lymphocytes. And of course, you got monocytes there as well. So those are all your white blood cells, but those are the percentages. But keep in mind, these are your largest and second largest amount by volume. So what we're seeing here, and I believe you have seen this in the lab, but what you see here is under a microscope what you would look at for, to identify not only the red blood cells, which are found in these areas of the slide or the specimen, but in fact your white blood cells. Neutrophils will always have this kind of multi-lobe nuclei, and you can see this designated by this is one nuclei versus another, so multi-lobe nuclei. And you can see this one, in fact, has 5 there, right? So that's always how you can tell what a neutrophil looks like. An eosinophil will have this bilobed nuclei. Basophil has these little tiny dots all through the white blood cell. A lymphocyte will have a very large nuclei, and a monocyte will always have this kind of U shape or even kidney shape. So sometimes it kind of looks like this, or sometimes looks like in fact, a kidney. and that's a monocyte. You can also look at the size difference, so you can see how the size difference is with each one in comparison. You can see if I erase the monocyte, much bigger. So just to look at that. Now I mentioned granulocytes, and those are the three that are considered granulocytes. Okay, these are all phagocytic. Neutrophils and granulocytes, in fact, are phagocytic, so they have the capability of consuming, infections like bacteria, or even consuming debris. Right. So when I always talk about a phagocyte, I always use Pac-Man as my example. And just like the game Pac-Man, the goal is to actually eat away at those little white circles, and then eventually the ghosts, right? When you change a certain color, right? So picture these as your little bacteria that are trying to invade your body. The neutrophils, eosinophils, and basophils recognize those as foreign, and that's key to understand that they recognize them as foreign first, and then they elicit some sort of attack. And this attack is phagocytosis, because once they consume them in, those are where the granulocytes actually play a role. Excuse me. So neutrophils, as I mentioned, are the most numerous, twice the size of a red blood cell by diameter. And now we talk about those granules. The granules in a neutrocyte have hydrolytic enzymes or antimicrobial proteins, and we would call those antimicrobial proteins defensins. There are many different types of defensins or proteins that are antimicrobial, so that can vary, but we do see that those granules can be either or. Now, when we talk about hydrolytic, enzymes, this is actually two words in one. Hydro means usually hydrogen, or sometimes water, in fact, and lytic means to actually break, right? If you don't know what that means. And actually, those enzymes are designed to break hydrogen bonds between, for example, proteins or other chemicals that are part of, let's say, a bacteria or a foreign object. So, in fact, when they phagocytize these chemicals or this, for example, foreign pathogen like a bacteria, and what they do is they have these vesicles inside of them, and they release those vesicles. They throw the debris, for example, in the vesicle, and then they release those granulocytes. Those granulocytes, and in particular the hydrolytic enzymes, can actually completely break down a structure like a bacteria, and actually kill the bacteria. So we call them bacteria slayers, and that's why I'm discussing bacteria, but they're very phagocytic to bacteria. Okay. Neutrophils, in fact, can kill microbes by what we call respiratory burst as well. So this is another option. And so the cell of a neutrophil can actually make bleach, in fact, hydrogen peroxide or hydrogen hydroxide, sodium hydroxide, which is bleach. Those are two oxidizing agents, and what they act as is actually steal electrons or take electrons, which actually break bonds in chemicals of let's say, the organism. So what about those antimicrobial defensins that we talked about before, and mentioned that those granules can be defensins also? The vesicle that a neutrophil takes in and traps the bacteria, let's say, inside of the neutrophil, the granules merge and get released in the vesicle, and we call that a phagosome. And those granules can actually pierce holes in the membrane of the microbe or the bacteria. So it's actually, quite interesting. The neutrophil, in fact, will do this, and eventually, if this is a bacteria, will eventually, and we'll fast forward here, swallow up the bacteria and form a phagosome. That, in fact, would be a phagosome. And then that phagosome can be released, can be interacted with granules, and those granules, in fact, get released inside the phagosome and actually can punch holes in the bacteria, thereby destroying the bacteria. So again, very interesting how some of these cells work, and that's your neutrophil. Okay, so an eosinophil, again, small percentage there, but an eosinophil about 2 to 4% there. It's got two lobes. It's usually the nuclei is a two-lobe nuclei, and an eosinophil. They have digestive enzymes. Those granules are digestive enzymes. And they usually work on a specific type of microbe or an organism, like parasitic worms. Now, what they also can do is play a role in allergies and asthma, because the release of those granules, for example, outside of the eosinophil, can actually act as a immune system regulator. So the release or non-release of those can act as a cascade to start a process of the immune system. And so we can see that in, for example, allergies and asthma, those conditions where you have an allergic reaction from pollen, let's say, and released from plants, flowering plants, or some sort of chemical or any other type of chemical that your body reacts, and in particular your respiratory system, that cause asthma. It's just kind of a spasm of your bronchi. Okay, so there's the picture of a eosinophil as well. A basonophil, okay, rarest one, the smallest percentage of the basonophil, and again, we're just talking about granulocytes. And those granules are a little bit different. Those are histamine granules. Now, histamine granules, when released, act as an inflammatory chemical. They act on the blood vessels, and in particular the capillaries, resulting in vasodilation. Okay, so if I am looking at a blood vessel, and here's my typical blood vessel, I haven't drawn, in fact, the layers of a blood vessel. But let's say this is a blood vessel, now this is a cross-cut view, so we're looking, we cut the vessel, we're looking inside of the vessel. So let's say this is the normal diameter of a blood vessel, and in vasodilation, when those histamine chemicals act on the red blood cell, we see an increase in the, in fact, diameter. Does that make sense? The diameter, how wide it is. Vasodilation is an increase of the blood vessel to allow more blood flow. Why blood flow? Because we can allow more white blood cells to the inflamed site. So one of the first cells to show up in an inflamed site, in a damaged site, would usually be those neutrophils, and then quickly by the other types of white blood cells. When the basophils arrive, they release these granules out in the bloodstream to cause more vasodilation in the area, so that more blood flow can go in, and then we can actually increase the amount of white blood cells to the area. That is what the function is going to be. Okay. All right, so onto the agranulocytes. So they don't have no granules, right? Do not have granules. And you've got two types, lymphocytes and monocytes. We'll talk about each one here. Lymphocytes, as I mentioned, the second most numerous ones by percentage, about 25% of those. Lymphocytes are usually found in lymphoid tissue. Now, this is part of the lymphatic system. Okay. And the lymphatic system, we'll begin discussing that after our first exam. That's our next chapter after blood vessels. But the lymphatic system is a kind of a holding area and works with the immune system, which is kind of what we're discussing here. And as far as blood is concerned, because we're kind of dipping into a little bit of immune system. But in fact, the lymphocytes are found in those lymphoid tissues. And what do I mean by lymphoid tissues? If you know what lymph nodes are and the spleen, those are considered parts of or types of lymphoid tissue. Okay, so really important to immunity, and this is actually one of those cells that they are the first line of defense towards the immune system. So they're actually run into usually if you see some sort of foreign body in our cells, they're the first ones that run into those through the lymphoid tissue. And so there's two types of lymphocytes, and we'll discuss more of this in the white blood cells, but these are T lymphocytes and B lymphocytes, known as T cells and B cells. T cells act against usually virus-infected cells, and tumor cells, but they also work with B cells, which form then plasma cells that produce antibodies. And antibodies are made by and secreted by plasma cells, which are really important for the immune system to function. All right. So that's a typical leukocyte there. Monocyte. This is the biggest by size, about 3 to 8% of that of all white blood cells are monocytes. Like other white blood cells, they are capable of leaving the blood circulation, going into tissues, and turning into what we call macrophages. And macrophages are just actively phagocytic cells that migrate into tissue or are found already in tissue. And they like to go to a location possibly where there's an infection or a breach of the immune system. They are important to mount an immune response, in fact. And this is an example of a leukocyte. Again, the biggest one as far as size is concerned. So again, here are all your leukocytes. Okay, so we mentioned lifespan of red blood cells, erythrocytes, previously in our discussion of blood. Leukocytes also have a production, a start, and a lifespan as well. So we'll start at the production side, or the making of white blood cells, and we call that leukopoiesis. So we use the word poiesis to be talk about making, but leuko means white blood cell making, right? So there are two chemical messengers that stimulate the production of leukocytes. Interleukins and colony stimulating factors, or CSFs. Those get produced by the body, and when secreted out, they actually stimulate white blood cell production. Okay. Those leukocytes originate from that hemocytoblast stem cell. That same hemocytoblast stem cell that actually creates the red blood cells can also be dictated to create white blood cells. So if you're a leukocyte, you fall under two pathways. Either you're going to be those hemocytoblasts will become lymphoid stem cells or myeloid stem cells. So here's your hemocytoblast. And you can be designated to become a lymphoid stem cell or a myeloid stem cell. The lymphoid stem cells will then become, eventually, lymphocytes. Myeloid stem cells will become all other white blood cells. And so we're looking at the granulocyte production. We see that there. We see how we turn the granulocytes. We've got after the myeloid stem cell becomes a myeloblast, promyelocyte, myelocytes, band cells, and they will become then a specific type of granulocyte. Agranulocytes also come from the myeloid line. And so it's a much shorter line, monoblast, promyelocyte, and then a monocyte. The lymphocytes come from the myeloid line, and they have an intermediate or a middle cell, and we call that a precursor. And so that precursor, the lymphoid stem cell, becomes the T lymphocyte precursor or the B lymphocyte precursor. And that gives rise to immature T cell, immature B cell. Now keep in mind that the immature T cells become mature in the thymus. And we'll visit this again in the immune system, but I would expect you to know this already. So make sure that you know this. The immature B cells become mature within the bone marrow, which is actually the site of the synthesis of all blood cells. So the B cells essentially stay behind, and the T cells travel to the thymus and become mature there. So we become mature T versus B. Okay. So here's the leukocyte formation. And again, you can see, in fact, if I'll increase this just a little, all blood cells become, or all blood cells derive from hemocytoblasts. And so on the white blood cell line, you have those that become lymphoid stem cells, and those become myeloid stem cells. The lymphoid stem cells become your precursor B and T lymphocytes. The myeloid side becomes those myeloblasts. And after they become a myeloblast, then they, in fact, differentiate into promyelocytes, and then they will become, through a process, eosinophils, basophils, neutrophils, or monocytes. Again, this is on the myeloid side. All right. So what I want you to know is, of course, the hemocytoblasts, lymphoid and myeloid stem cells, and which lineage it becomes, right? Which white blood cells, what lineage are they from? So T cells and B cells come from the myeloid stem cell line, and the monocytes, neutrophils, basophils, and eosinophils comes from the myeloid. So I'm sorry, lymphoid stem cell from the B and T, and the myeloid from the rest of the white blood cells. Okay, so let's look at this homeostatic imbalance here. So many of these types of hormones that stimulate the creation of white blood cells and red blood cells, this being the hormone to stimulate for the production of red blood cells, and this one for white blood cells, can be used clinically. So why would you use this clinically? Cancer patients, for example, receiving chemotherapy. And if you know about chemotherapy, it actually strips your natural white and red blood cells, including your stem cells in your bone marrow. So that can be a reason why you would use that. Or conditions like AIDS patients, in which it is a viral infection that attacks the immune system, kills those cells of the immune system, and so you need a stimulation of more white blood cells, for example. So we've got a lot of different leukocyte disorders, right, out there. And then in particular, they fall under, most of them fall under these groups here, right? Leukemias, mononucleosis. This is the overproduction of white blood cells, or a low white blood cell count. We call this leukopenia. Okay. Leukemias, this is the cancerous condition. And what we see is there's a single abnormal cell. The abnormal cell has lost the ability to regulate when it produces, which means when it undergoes mitosis or cell division. And so the mutation has caused it to actually divide uncontrollably. And so what is happening is that stem cell has grown uncontrollably, divided uncontrollably. And so we have many clones of that abnormal cell. Hence the overproduction of white blood cells. So when you talk about a leukemia, you can be specific as far as what type of leukemia it is, depending on what white blood cell clone it produces. So, for example, I know a lot of families are familiar with myeloid leukemia or multiple myeloma. Multiple myeloma is because the clone is the myoblast. So that myoblast is one of those is grown uncontrollably and hasn't stopped. It's lost its ability to regulate. So it undergoes rapid mitosis consistently. A lymphocytic leukemia involves the lymphocytes. So it talks about what we're talking about here is one of the precursor B and T cells that has grown, that's divided and hasn't stopped. So an acute leukemia is usually because the clone or the abnormal cell is a stem cell. It becomes a stem cell, or if it is the stem cell, it's acute. Quickly advancing, because obviously the stem cells will grow uncontrollably. Usually affects children, so that's the type of leukemia you see in children. The chronic one usually involves later cell stages. Some of those precursors or the myoblasts or some of those, right? This is usually more prevalent in older people. So, for example, multiple myeloma will usually affect older folks. Your chances are greater after a certain age, 50 or so. And actually, most multiple myeloma cases are after, I believe, 65 or 70. Okay, so let's look at the other one here, this overproduction. This is infectious mononucleosis, and in fact, this comes from a virus. The virus is called Epstein-Barr virus. This virus is also commonly known as the kissing disease, although kissing is not the only way you can contract mononucleosis. That can also be from sharing cups or water or other saliva exchanges And that could be saliva on a surface and you touch it with your hand and you put your hand in your mouth. That's how you can get mononucleosis, right? So usually the Epstein-Barr virus is going to involve the lymphocytes. So they involve the lymphocytes and invade the lymphocytes and cause those lymphocytes to increase in size and then also increase in number. So originally we thought it was the virus only invaded monocytes, so that's why we call it mononucleosis. But in fact, it invades lymphocytes. So here's some of the symptoms there, but mononucleosis usually runs its course about 4 to 6 weeks. And again, it's prevalent in, well, not prevalent, but you find this in younger adults. Okay, so now into platelets, right? So this is another type of, type of blood cell. And I wouldn't call this a whole cell. I would call this a portion or pieces of the cell. So we've discussed RBCs, we've discussed WBCs, and now we're discussing another major type of cell, and I would say cell piece, and those are platelets. Platelets are actually fragments or pieces, talking about pieces of a larger cell, and we call that larger cell a megakaryocyte. Megakaryocyte. So a megakaryocyte, in fact, the pieces of a megakaryocyte that we call a platelet actually has several chemicals which actually play a crucial role in the clotting process. And here are some of the chemicals here. Those, when released by the platelets, actually help facilitate the further clotting process. And what is clotting? We're talking about clotting of blood, or clotting of a blood vessel that's been broken. That clot is actually a temporary plug. We're actually plugging the hole to prevent further blood loss and to give the body enough time to repair the blood vessel. Platelets and their forming come from another hormone. We call that thrombopoietin. Thrombopoietin is a hormone. That hormone, when released, stimulates platelets to actually begin the process of forming the entire cell and then forming the platelets that you see. It's formed in the myeloid stem cell line from a stem cell we call a megakaryoblast. That's another type of stem cell. So platelets have a very short lifespan, about 10 days, and they degenerate in that lifespan and then have to be replenished, which they are. And you got about 150 to 400,000 platelets per milliliter of blood in your body. So here's the step process of developing platelets. Here's your hemocytoblast. And there's different stages, stage 1 through 4. But you see how the size of the cell grows bigger and bigger. And then eventually this megakaryocyte breaks itself into pieces. And those pieces we call, in fact, platelets. Okay, so this table here in your book, table 17.2, summarizes all of your blood cells. So we see at the very top your erythrocytes, all of your leukocytes that we discussed here, and your platelets there. Okay, so I want to talk about this process we call hemostasis. Hemostasis. Hemostasis is known as a step reaction for the stoppage of bleeding. So this is the step process of this stoppage of bleeding. This also involves clotting, which also involves platelets. Right? So in the part of platelets, which is in that part of platelets doing their job, as we mentioned, platelets also release chemicals. Those are also considered clotting factors. Those chemicals actually help to uh, continue the process of this clotting. There are three main steps in hemostasis. Step 1, vascular spasm. Step 2, platelet plug formation. And step 3, coagulation, clotting. And in step 1, when we talk about vascular spasm, we're talking about the blood vessels actually constrict. Instead of dilate, they constrict. That spasm is triggered by injury to the vascular smooth muscle, chemicals that get released by the endothelial cells that line the blood vessel, that when they're broken, release those chemicals. They cause the spasm, or reflexes and pain. So if the nervous system dictates that as pain, then we can see that there as well. So this gets more effective in smaller blood vessels, and so this vasoconstriction can reduce blood flow. So that's step 1, is to actually start vasoconstriction. That vasoconstriction can stop then or slow down the flow of blood, which is important if you want to reduce the amount of blood you're losing. Vascular spasm is important. All right, so a little bit about a blood vessel, because that's our next chapter. But if this was my crosscut of my blood vessel, in fact, I would have different layers of my blood vessel. Try to use different colors here to designate. For example, each of the layers. So let me go back here. We would consider this first layer tunica. Tunica is a layer, right? We call this the tunica externa. This layer right here between the red and the black, between the black and the purple, we call that the tunica media. And then your last layer between the purple and the blue, there is the tunica interna. The tunica media is an important one because the tunica media is mainly made up of smooth muscle. And that smooth muscle is important when we talk about, or when it's discussed, when we talk about injury of smooth muscle there. So if that blood vessel breaks some of the smooth muscle, then the release of the injury of that causes a vascular spasm. The tunica interna is where you find the lining of endothelial cells found in the tunica interna, which is a line of epithelial cells that lines the inside wall of the blood vessel. So that's what they're talking about there. Step 2 is platelet plug formation. So this is where platelets start to stick. The collagen fibers that get exposed when the vessel breaks or is damaged. So when the vessel breaks, collagen fibers are kind of opened, and platelets stick to those collagen fibers. And then platelets begin to stick to each other as they go through the bloodstream. Platelets swell when they become activated. They become sticky and spiked, and they release these chemical messengers. This one, ADP, adenosine diphosphate, released by platelets, causes more platelets to stick and more platelets releasing their contents. Serotonin and thromboxane A2 enhance vascular spasm and then recruit more platelets to the area. So if you hadn't figured out, we are talking about positive feedback in homeostasis. So this causes the sticking of more platelets, the release of more chemicals, causing more platelets to stick, and on until we plug the break. Step 3 is coagulation. And in coagulation, we have blood clotting. The platelets get reinforced with fibrin, which is a type of connective tissue. And this coagulation, it has its 3 phases, and we'll look at it, but I don't want you to really be too concerned about each of them, one of those phases. In your book, figure 1713, you see the 3 major events of homeostasis. That's step 1, step 2, step 3. And I went ahead and put those in a step process as well, so you're able to kind of go back and forth and look at it in each individual step. So coagulation occurs in either the intrinsic or extrinsic pathway. In the intrinsic pathway, we call this intrinsic because those clotting factors are found within blood, and it triggers this activation of platelets and collagen, and then the clotting to occur. The extrinsic pathway are factors that are located outside of blood, and that's usually because the tissue's been damaged. So we find that from the outside of that's not blood tissue, the part of the blood vessel that's gotten damaged. And both of those pathways actually work together to speed up clotting. And so you see the intrinsic versus extrinsic pathway. Okay, in the intrinsic pathway, you see that the endothelium layer, the inside layer, gets broken, causes the platelets start sticking, and this is kind of the inside pathway. The extrinsic pathway is from the cells that get damaged, and we're talking about smooth muscle and sometimes the external part of the external part of the blood vessel. Both of which then begin the process of finally coagulation or blood clotting. And so we see this step process, and there's several different, so we call this a cascade, right? Like the domino effect. In the fact where we see the release of something causes then another release of something else and activates another chemical, which activates another chemical. And all of this here activates this cascade, this clotting cascade. You do not need to be familiar with all of those there. You just need to know that there's 2 different pathways, and that the clotting is part of that step process of temporarily plugging up a blood vessel that's been damaged. And so all of this discusses a little bit more into that process there. So I want to show you this picture that you see in the book, but in fact, this is the scan electron microscope of the erythrocytes getting trapped by this fibrin mesh. This fibrin is connective tissue, and we start to see this mesh form, which is part of the process of this plug, if you will. Those are all your clotting factors, in fact, and these you do not need to know, but they show you that there are 13 clotting factors, and you can see where they come from. Most of them come from the liver. In fact, they get made by the liver. And so those are found, for example, circulating in blood, and they're inactive until one of those chemicals that is activated causes the other one to activate. And so we see this important process. Okay. So the other part I wanted to just mention to you is talking about, for example, the human blood groups which we've discussed that, or you've discussed us in lab. I believe lab, you've already taken a blood test and looked at the clotting factors in blood, the antigens and the antibodies that cause clotting, so we can look at blood groups there. And so you've seen this, so I feel comfortable. There is some information on this if you'd like to go over that. And you should be going over that in your book. There's extra information for that in the PowerPoint. But again, you've discussed this with Ms. Gonzalez in lab, so I'm okay with us not covering that today, or at least in this presentation. And this is what you see, right? ABO blood types. Okay. I think we've covered everything from where we stopped in lecture to this point. And again, make sure you go through the overview to make sure that you covered anything else that wasn't getting covered here or in lecture.